Hello students. This is the module on new drug development in allopathy medicine. Dr. Rakhi Tripathi has prepared the module and I, Dr. Ashwini Karve, would be presenting the same. Our session objectives will be define new drugs and get aware about the regulatory guidelines or framework followed in India for new drug development. Mention the stages for new drug development, namely preclinical and clinical development. Discuss the objectives of preclinical, that is, animal testing of drugs. So, with these objectives in mind, let us further have or add two more objectives. Discuss clinical development, which occurs in phases as phase 1, 2, 3, and 4, highlighting their objectives, requirements, and ethical consideration. In homeopathy, there is drug proving protocol, wherein the drug is directly tested in healthy volunteers. Now, this differs in allopathy. Earlier in allopathy, more than 70 years ago, drugs which were used to treat diseases were actually discovered either by chance or by enormous efforts to find a new drug. Today, this drug discovery is changed to drug development, which is planned, regulated, scientifically and ethically robust process which is country specific and conducted by pharmaceutical companies responsible for manufacturing and sales of the drugs in the market. Drug development involves several steps, which can be summarized as follows. First step is drug designing. Based on the insights gained from existing drugs, the new drug formulation can be designed either by target identification as gene or genome sequencing, which involves identifying and sequencing the genes or genomes of interest to understand the biological target for potential drug development. Target discovery and validation. Researchers identify specific molecular targets such as proteins that play a role in the disease or the condition being targeted and then validate to ensure their potential effectiveness in treating the disease. The new drug molecule is searched as chemical compound that can interact with the target and potentially become the basis for a new drug. These leads are then validated and optimized to enhance their efficacy and minimize any potential adverse effects. Later, this lead molecule is tested in animals called as preclinical studies, which help to gather important data regarding dosage, toxicity, and potential interaction. Once this molecule is proven to be safe and effective for the disease under consideration in preclinical studies, it is further tested in human beings in phased manner called clinical development. Subsequently, when the drug has successfully completed clinical trials and received regulatory approval, it enters the manufacturing stage, where it is produced in large quantities following strict quality control measures and then distributed to healthcare providers, pharmacies, and marketed. It's important to note that the drug development process is complex and can vary depending on the specific disease. The steps provided here offer a general overview of the typical drug development journey. The regulator, which controls new drug development, manufacturing, and distribution in India, is CDSCO, that is Central Drug Standard Control Organization and Monitoring Drug Safety in India, which has Drug Controller General of India, that is DCGI office, which would review the new drugs. DBT-RCGM, that is Department of Biotechnology Recombinant DNA Advisory Committee, reviews and approves research proposals involving genetically modified organization as drugs. While GEAC, that is Genetic Engineering Appraisal Committee, evaluates and approves genetically engineered organisms and products. DGFT, that is Director General of Foreign Trade. These facilitates the import and export of pharmaceuticals, the issue licensing authority, and regulates trade-related aspects in the pharmaceutical sector. The Drug and Cosmetics Act is a key legislation in India which governs drugs, cosmetics, medical devices and related matters. It was first issued way back in 1940 and then revised in 2005 to align with the international standards. The Act provides legal support for good clinical practice, that is GCP guidelines, ensuring ethical and scientific conduct of clinical trials. It has contributed to improving the quality of clinical trials by mandating compliance with GCP guidelines, including ethical principles and robust study designs. As stated earlier, regulatory oversight is provided by CDSCO and its department headed by DCGI. The Act has undergone amendments to keep up with advancements and challenges in the healthcare sector. The latest regulation which is applicable are the new drug and clinical trial rules of 2019, which outline the requirements and guidelines for obtaining permission to import, manufacture and sell new drugs as well as for conducting clinical trials in the country. These rules were introduced to streamline and expedite the drug approval process while ensuring the patient safety and ethical conduct of clinical trials. One important document released to these rules is Form 44, which is used to submit information regarding the import, manufacturer or sale of new drugs and clinical trial applications. Let's then look as how we can define new drug. A new drug is defined as a new substance of chemical, biological or biotechnological origin used for prevention, diagnosis or treatment of disease in man or animal and has not been recognized in the country as effective and safe for the proposed claim. A drug which is already approved and available in the market can be considered new for modified or new indication, dosage forms and route of administration or as a fixed dose combination of two or more drugs. Let us now focus on preclinical development, that is, testing of new drugs in animals. The primary objective of preclinical studies are to gather information about a drug's efficacy and overall safety profile before advancing to human clinical trials. These studies are essential for fulfilling regulatory requirements for drug registration and approval. They involve in vitro experiments, animal studies, and other non-human testing methods to provide valuable data on drug behavior, dosage, toxicity, and potential interactions. Further, to elaborate preclinical studies encompasses several types of experiments conducted to assess various aspects of drug, such as specific pharmacology studies. Now, these studies are performed in animal models that simulate the disease or condition being targeted by the drug. They aim to evaluate the drug's effect, such as its mechanism of action, therapeutic potential, and effectiveness in treating the specific disease. Safety pharmacology studies. Essential safety pharmacology studies, which are conducted to assess the potential adverse effects of the drug on vital physiological functions, and follow-up safety studies may be performed to gather additional data. Pharmacokinetic studies. These studies investigate the drug's absorption, distribution, metabolism, and excretion properties. These studies provide clinical insights into how the drug behaves within the body. Toxicity studies. These studies would assess the potential adverse effects and toxicological profile of the drugs when given in excessive amounts that will act as actually poison. These studies will help identify the drug's safety margin, and it would also inform the dosage recommendations. Safety pharmacology studies in preclinical testing investigate the drug's impact on vital organ functions such as central nervous system, cardiovascular system, and respiratory system. These studies help identify potential risk and guide decision making in the development of the drug. Toxicity studies are a crucial component of preclinical testing. Assessing the potential harmful effects of drug candidates is an important aspect. Standard tests include acute toxicity and chronic toxicity evaluations. Acute toxicity studies would determine the adverse effect to a single exposure, while chronic toxicity studies would assess long-term effects after repeated administration. Special tests may include female reproduction and male fertility studies to examine any potential reproductive toxicity. Local toxicity studies investigate the drug's effects on specific organs or tissues. Additional tests such as allergy or hypersensitivity testing, genotoxicity, and carcinogenicity assessments are also performed. This, apart from proving new drug to be effective in simulated disease conditions in animals, equally important is to prove safety and calculate the amount of drug that will work as poison or toxic. Hence, toxicity studies also become an important aspect. These toxicity studies would have specific objectives such as estimate safe starting dose of a drug for human clinical trials and determining the highest dose that can be administered without causing significant harm. Researchers can establish an approved initial dose for testing in humans. Assess target organ toxicities. This helps identify any potential organ-specific toxicities that should be closely monitored during subsequent clinical studies to ensure patient safety. Assess potential reversibility. Now, these studies would provide insights whether the adverse effects are reversible after discontinuing the drug. Researcher can better understand the potential risk and outcomes for the patient. Further, the assessment of genotoxicity and carcinogenicity is of paramount importance. Genotoxicity studies would assess ability of the test drug to cause DNA damage or mutations, while carcinogenicity, these would involve long-term dosing of animals, usually 18 to 24 months, to observe the potential of the drug to induce cancer. When conducting preclinical studies, there are minimum requirements that must be met to ensure ethical and reliable research. The study protocol must be reviewed and approved by Animal Ethics Committee. Well-maintained and registered animal house that meets appropriate standards for housing, care and maintenance of laboratory animals is a must. This ensures the well-being of animals and compliance with animal welfare regulations. And preclinical testing is conducted by veterinary doctors and qualified researchers. Once preclinical studies are successful, chemical and pharmaceutical development, including pre-formulations, formulation development with assured quality is performed, and the drug is synthesized in bulk. These processes aim to create safe, effective, and high-quality drugs for patient use. So after successful completion of preclinical studies, new drug application, that is Form 44, is filled at DCGI office to now conduct testing of drug in humans. Appendix 1 outlines the data that needs to be submitted for clinical trials of new drug. It includes a brief description of drug and its therapeutic class, full chemical and pharmaceutical information of the drug, including its composition, structure, and manufacturing process is required. The regulatory status of the drug in other countries should be provided. Full prescribing information, which includes dosage, administration, indications, contraindications, and adverse reaction, is also required. These data are crucial for regulatory authorities to evaluate the safety, efficacy, and appropriate use of new drug in clinical trial in humans. In humans, the drug is tested in phases, and in each phase, experiments are conducted in human which are called clinical trials. They are defined as a systematic study of new drugs in human subjects to generate data for discovering and or verifying the clinical, pharmacological, adverse effect profile with the objective of determine safety and efficacy of the new drug. Let us now go through the journey of a clinical trial. Every clinical trial to be conducted requires writing a detailed protocol describing how the drug will be tested and what will be measured. This protocol, before execution, needs regulatory approval from DCGI, and at the site where it will be executed, approval from Institutional Ethics Committee is needed. After the study is conducted, the data collected is analyzed and submitted as trial report to the regulator office. Clinical trials are conducted by qualified physicians. Blood samples collected are processed at labs which are GLP certified and compliant to ensure the generation of high quality and reliable test data related to the safety of the new drug. Use of overseas lab requires special permission. Protocol amendments, that is, if you're going to change the protocol after it has been approved, then again you need to resubmit it to get the permission from the Ethics Committee as well as from the DCGI office. Phase 1 clinical trials are the first evaluation of a new compound in humans who are healthy volunteers. Objectives are to evaluate pharmacokinetic profile as well as the safety and tolerability of the new drug. Also, the safe dosing range can be found out. Phase 1 trial design is of dose escalation study with preferred route of administration. In addition, effect of single or multiple drug dose, preliminary effect of food and other drugs on this new drug is also assessed in healthy volunteers. In phase 1 trials, the number of normal healthy volunteers is less because this is the first time a human is being exposed to the new drug. So it ranges from around 20 to 100. The duration of study is around 6 months to a year, and investigator is a clinical pharmacologist. After successful completion of phase 1 trial, that is, new drug is safe in healthy individual, the phase 1 trial report is submitted to DCGI office and permission to conduct phase 2 trial is sought. After the drug permission from regulator and ethics committee for phase 2 clinical trial are conducted. In this place or in this phase, the drug is administered to a large group of patients with the target disease or condition to assess efficacy, optimal dosage and further evaluate its safety. Hence, phase 2 trials are also called as therapeutic exploratory trial, as the researcher attempts to find the dose range in patient for the given disease. Objectives are to evaluate safety and efficacy, determine dose and regimes, evaluation of potential study endpoints, and identify the disease subtype for which the drug is effective. This therapeutic exploratory trial, that is phase 2 trial, are feasibility studies which evaluate the efficacy, dose response rate and response duration of the new drug, coupled with, of course, safety monitoring studies in several 100 patients. Duration of this phase 2 trial is several months to 2 years, and clinical investigator or clinical pharmacologist would conduct such a trial. After successful completion of phase 2, again, regulatory and ethical approval is sought. Phase 3 trials, which are therapeutic confirmatory study, that is, it would confirm how well a new drug treatment works after it was shown in early phase clinical trial that it might be beneficial for the patients are conducted. Phase 3 clinical trials include testing the new drug on a large scale with broader patient population to confirm its efficacy, monitor the side effects, and compare its effectiveness against existing treatments or sometimes inert substances called placebos. To confirm therapeutic benefit or effectiveness within therapeutic dose range and to check the best dosage form, the study design can be open label, single or double blind with placebo or active comparator. These phase 3 trials are conducted in specific patient population for whom the drug is intended. Efficacy and safety data in large number of patients is generated. The data from such trials often provide information for package inserts and drug labeling later on. Phase 3 trials are conducted in 100 to several thousands of patients. Duration of study is around 1 to 4 years. An investigator is usually a clinician. After successful completion of phase 3, the pharmaceutical company now does new drug application for marketing. The DCGI office will grant marketing approval only after it's proven that the drug is beneficial for the patients with acceptable adverse effect profile. The launch of the drug, that is, coordinated efforts to debut a new product to the market and make it available for the public is done. Phase 4 studies begin after the drug is marketed. For these studies, also like earlier phases, permission from DCGI and ethics committee is required. The objective of phase 4 is to provide additional details or additional information about the medicine, especially the safety profile. Study design here would be open label, single or double blind. Phase 4 studies include post-marketing surveillance, wherein large number, that is around several thousands of patients are enrolled. Investigator is usually a clinician. In phase 4, the focus is more on adverse drug monitoring or periodic re-evaluation of safety, as the drug which was tested in several hundreds patients is now taken by several thousands of patients. This is done for the drugs with various drug dosages, different duration of drug treatment, interaction with food and or other drugs, and in addition, different age groups and patient types. While conducting all these trials under different phases, it is important that all these trials are conducted ethically, causing minimal harm to the patient and is beneficial to the society. The two pillars of ethics include, first, the ethics committee approval, and second, voluntary written informed consent taken from every patient or healthy volunteer who is a part of the study before they participate in the trial. Ethics committee of the institution will review the project and check whether it is in accordance with the existing guidelines. Thus, to summarize, drug discovery extends from 2 to 5 years, wherein target selection, lead finding, and optimization is done. This is followed by preclinical development, that is, testing in animals for almost 1.5 years to evaluate efficacy and safety of the new drug. Then later, once the drug is completed preclinical study, the drug enters clinical development or clinical trials, which would stretch for around 5 to 7 years. This is of different phases, as we already discussed. Phase 1 trials focus on tolerability and side effects, but they are done in healthy volunteers. Phase 2, small scale trials in patients, but now to assess efficacy and the dosage form. Phase 3, they are usually given or usually done in large scale patients. They are controlled clinical trials. And after phase 3, the regulatory approval and marketing of the drug is done, and then after that, phase 4 trials are conducted. So phase 4 trials include post-marketing surveillance for continued safety profile of the drug. Thus, every new drug that you see in the market is the extensive effect over 10 to 12 years of the pharmaceutical company and the clinicians who perform the clinical trials. These are few of the references. If any of you are interested more in learning about how the new drug is developed, you can go through these references. It will definitely help you. Thank you so much.